Okay, so now we're going to look at a nonsense mutation. So again, we have the normal DNA up here. We've got our mRNA that was transcribed from our template strand. We've got our string of amino acids that we figured out by looking at our codons in our mRNA, and that makes our protein. And then down here, we've got our mutation. So for this example, we have an A added here instead of a T during replication. And so when we look at the complementary DNA strand, we have a T here. Okay, so we've got our mutation here in yellow. And then that template strand is red during transcription. And so if we look at the complementary mRNA nucleotide to A, we get U. Okay, so now if we go along and read these codons in mRNA, and again, we need our genetic code. So we're going to look up AUG, and we'll see that methionine is added. And then if we go here and we look at UAG in the genetic code, this is actually a stop codon. So that means that during translation, all of the pieces fall apart. So the ribosome falls off, the mRNA falls off, the tRNAs fall off, and we're left with an amino acid strand. But because this stop codon occurred too early, this makes a short strand of amino acids, and generally the protein is not able to function at all because it doesn't have that correct strand of amino acids, and so it can't work. Okay, so a nonsense mutation is when we have a change in the DNA that causes a stop codon in the mRNA.